thank you very much, and bon dia, everybody. I'm Romain JEANTET, as Guy and Antonio kindly stated before. I'm the vice head of Science and Technology of Milk and Egg, the research unit that I share with Thomas, which is based in Brittany, actually. And this is the place where Guy and Antonio did their PhD more a long time ago. And today I will give you an overview of the work we conducted with the colleagues that are specified here: Lucas Lanotte, Ming Yu, Shong Wang, Florian Gaucher, Maïchandra Patil, 4 PhD students, and my direct colleague Lucas Lanotte and Gaël Tanguy, on this topic of drying of whey constituents and products from physical and biological mechanisms to breakthrough innovation. So just to give a short view of how we position, our research aims at answering the context of the development of dairy powder products, among which the key facts are, of course, on the one hand, the massive growth of infant milk formula, IMF, that represents nowadays 2 million tons a year at a global scale. And on the other hand, the protein isolate market, in relation to nutritional products development. These products should present controlled properties and in a short term incorporate live probiotics, given the growing interest for microbiota concern. And of course, they must also integrate environmental and cost concern to position themselves as sustainable and competitive products. We have 3 research topics. So the first is the understanding of the particle formation mechanisms, which is very important. The second one is the identification of the key parameters that rule the powder properties and evolution. And general topics, which is kind of transversal, to explore new technological concepts for technological breakthrough innovation. So to address these topics, we implement a strategy that is based on simplified and controlled models regarding, for example, the dairy colloids. We come back on this, or bacteria. Picturing the phenomenon at different scales. The combination of disciplines, which is absolutely necessary, from biology to soft matter and chemical engineering in a multidisciplinary approach, and as much as possible in close partnership with dairy companies and in the frame of PhD projects. And at the bottom, you have pictures of the PhDs that were implied in the work I present today. So to illustrate these issues, I will shortly give you a contribution on the 3 research topics. First, by exploring particle formation to control the infant milk formula properties. The second, by triggering the protection mechanisms to maximize survival of bacteria. And then the redesigning of the process for the production of whey permeate powders. So to study the particle formation, we use a multi-scale approach that we develop on pure milk proteins, so the whey proteins that I will call WP here. The micellar caseins that I call MC, and this protein can be studied single or mixed together. And the first stage of this strategy is the following of the drying of a single droplet, either 2-dimensional or 3-dimensional, that is deposited on a pattern. And no matter the drying of a suspended single droplet is different from real spread drying, if you want to understand the spread drying, you first have to understand the drying of a single droplet. And so we developed the specific experimental setup to control and study the drying kinetics and record in situ deformations of the single droplet during drying to finally measure some mechanical properties of the dry particles. The second stage of the strategy consisted in challenging the robustness of these results on monodispersed flying droplets. And as the droplets are monodispersed, they are encountering the same control drying history and show the same characteristics. We can collect in this stage homogeneous and big enough samples to analyze representative particle properties. And last, we validate and confirm the results on an industrial scale. So with this method and this approach, we extensively studied the drying of dairy colloids, and you have here some references in blue on the top left that we published among the past years. There will be some more in the next slide. And we found that almost regardless of the drying kinetics established by the drying temperature, the size of the droplet and the drying scale, specific signature of whey proteins and the micellar caseins influence the particle formation, final shape, and properties. Shortly, both proteins showed the same drying stage. The first is the free evaporation of water from the droplet surface at constant rate and simultaneous isotropic shrinkage. The second, once the sol gel transition concentration is reached at the interface, a skin is formed, impeding the free evaporation of water. And we found that the sol gel transition is reached earlier for micellar casein in accordance with the phase diagram of the both proteins. Then the mechanical properties of the skin condition the final particle shape. And we have two particle shapes, as you can see here. Spherical hollow particles at the top for WP, whey proteins, and collapsed ones for MC proteins. And regarding the particle properties, the WP had a hard sphere-like behavior. That is to say that when the drying stress reaches the yield stress of WP, the shape remains spherical, but the occurrence of fracture reflects a brittle plastic material that is fragile. And on the other hand, the micellar casein behaves as a ductile, deformable plastic material. That is to say that the micellar casein has a sponge-like microgel behavior, not only at the molecular scale, but also at the droplet scale. So the question is now, is the skin composition representative of the bulk in the case of mixes? And in order to track the localization of MC and WP at the interface, we realized an elemental analysis of calcium as a tracer of micellar casein. So the figure here on the left shows the internal-external skin of the droplet that we fracture when it is finally dried. And on the right, it shows the internal-external skin calcium concentration as a function of the WP over micellar casein ratio, which is decreasing from left to right. So on the left, you have from left to, sorry, from right to left. Sorry. On the right, you have pure WP protein. And on the left, you have pure micellar casein in the bulk. And as you can see, the external skin surface, that is here the yellow bars, are overrepresented by whey protein, means that we have a low calcium concentration. And even when the micellar caseins are major in the bulk, which is the case, for example, for the mix 2080. You have a major composition in micellar casein. So now the question is how explaining such behavior. And in order to explain this, we realize some scanning electron microscopy that showed whey protein segregation at the interface. Indeed, the skin here that you can see, the external part, showed homogeneous density and porosity that is characteristic of whey protein. However, once we calculated the Peclet number, and the Peclet number is the ratio of evaporation, so from the internal to the external, over diffusion from the external to the internal. So when we calculated this Peclet number, we found much lower value for WP, 6, with comparison to micellar casein, 100. And then the evaporation should favor micellar casein accumulation at the surface, which is not the case. So in order to interpret our results, we used an interesting physical approach that was proposed recently, and it's called the small on top theory. And this theory makes it possible to model the diffusive cross interaction by taking into account first an osmotic pressure ratio, which is derived from the colloid size, and is called alpha, the Peclet number that I already mentioned, and the volume fraction occupied by the colloids, phi. And we found the value more than 1, as it is shown here, meaning that the significant osmotic pressure tends to accumulate smaller colloids, here the whey proteins, on the external part of the skin in agreement with our results. So all these results and study shows that you have a limited impact of what we call the diffusiophoresis, which is the molecular drive to interface due to diffusion, except when the whey protein over micellar casein ratio is lower than 2080. So this is a quite theoretical part, but I will propose you other parts more applied in the following of this presentation. And then to follow, let's move to a new process, which is consists of triggering the protection mechanisms to maximize the survival of bacteria. So the typical processing steps of probiotics spread drying are the sterilization of the growth medium, as you can see here, the separation and resuspension of the bacteria in a drying medium before spread drying itself. And so far, 3 strategies have been used to improve probiotics viability during spread drying. The first is the microbiology strategy, for example, by selecting resistant bacterial strains, or inducing cross-protection via sublethal stresses. The second is the material strategy, using protective food-grade materials as drying matrix to protect bacteria during drying, for example, proteins or maltodextrin. And the third one is the process strategy, optimizing drying process in order to moderate the drying conditions. Most of the time, these 3 strategies are studied separately without consideration of the whole process. And so our question was, is it possible to jointly consider the 3 to improve the viability of dried probiotics? In this case, this perspective, the microbiology approach, we know that the cross protection is often used, and as it is worked, and it consists of treating the bacteria, for example, with a sublethal osmotic stress to induce stress tolerance. And on the other side, on the right, from the processing approach, the pre-concentration of the feed materials before spread drying is mandatory to decrease the drying temperature, because the lower the dry matter, the higher the drying temperature. So our strategy was to combine the 3 strategies together by using a concentrated material as both growth and drying medium, and that we called at this moment a 2-in-1 medium, so both for growth and spread drying. It makes a simplified process, of course, as intermediate steps such as the separation and the resuspension are skipped. And at the same time, it lowers the contamination risk, and the probiotic metabolites that may be generated in the growth medium will be retained in the final powders, and energy saving should be expected. So we worked first on probiotics growth on the sweet whey medium, testing sweet whey recombination from 5 to 30% weight dry matter. And we found that on the one hand, increasing concentration makes the nitrogen non-limiting, which is beneficial for the bacterial growth. But at a certain stage, increasing concentration induces more and more osmotic stress, which inhibits the bacterial growth. So finally, we found, surprisingly, it was not what we thought at first, that 30% weight dry matter concentration was an optimum regarding the bacterial population yield. We then tested the two concentration in drying. So we used Lactobacillus casei BL23, which is a probiotic strain, and we grew Lactobacillus casei directly on the medium, and then we spread right directly the sweet whey culture and simultaneously checked the scale-up feasibility of this 2-in-1 process from the lab scale to pilot scale with regard, as you can see, both to fermentation from 2 liters to 500 liters and spread drying, the GA minor pilot on the left, and our Bionov workshop pilot, which is a multi-stage dryer that present a spread drying evaporation capacity around 100 kilograms per hour, which is representative from the industrial scale. So we implemented the two sweet whey dry matter growth at the lab scale, 5 or 30% dry matter, and two different schemes at the pilot scale. The first one that you can see here is a single stage. It's the same, in fact, as the minor stage, with same inlet outlet temperature than at the lab scale, so on the left. And on the right, the second one, which is multi-stage, with a belt drying and feedbed drying step. And these two additional steps made it possible to lower inlet-outlet temperature down to 127 and 47 degrees respectively. So the powders were then collected and analyzed. And here are the bacterial survival after drying results. The column that you can see here refer to Lactobacillus casei concentration and the solid curve to its survival. So we can see the bacteria survival significantly increased with the sweet whey concentration, and the 30% sweet whey dry matter medium led to 70% survival, while the survival was almost zero for the 5% dry matter sweet whey. Moreover, there was no significant difference between the results obtained at lab scale and pilot single stage scale, which validates the scale-up feasibility of this 2-in-1 process. And last, we showed that coupling the 2-in-1 process with the multi-stage drying scheme provided a 75% survival. So we were very excited, and we patented this process. And the question, our question at this moment was that what protects probiotics indeed. So to answer this question, we demonstrated that growth on 30% sweet whey led to upregulation of osmoregulation and stress proteins. The osmoregulation proteins is, for example, the glycine-betaine transporter, and stress tolerance proteins, maybe, for example, cysteine synthase, responsible for oxidative response. Plus significant accumulation of compatible solutes such as polyphosphate, glycogen, and trehalose. Trehalose is known to protect bacteria from extreme desiccation stress. All of this resulting finally in an enhanced multi-stress resistant phenotype. And you can see on these three graphs the results of Lactobacillus casei survival after different challenge tests. So from left to right, heat challenge test, acid challenge test, and bile salt challenge test. And you can see also that we obtained a higher survival when Lactobacillus casei was grew on 20 to 30% sweet whey. So it was very in accordance with this higher resistance towards spread drying. And moreover, we demonstrated that this was a generic trend that was also the same. It was the same for Propionibacterium and Bifido strains. And all of these strains grow well in 20 to 30% sweet whey, which is not expected at first. And the survival rate after spread drying may reach up to 100%, for example, in the case of Propionibacterium. We cannot, interestingly, if I want to make a small joke, that these results are in accordance with the word of Nietzsche, that said that what doesn't kill you makes you stronger. And to go further, we try to tune the osmolar adaptation to increase and control the probiotic survival. So the question was, can we tune this osmolar adaptation to enhance survival of, for example, Propionibacterium fraudenreichi? And to address this question, we grew the Propionibacterium fraudenreichi on different media, so namely whey, UF permeate, EL, and EL plus lactose on this figure from left to right. And all these medium are differing by their lactose over non-protein nitrogen NPN ratio, that is to say carbon over nitrogen ratio. So whey and UF permeate, as you know, mostly contain lactose, whereas EL contains NPN. These media were added with sodium chloride in order to reach a constant osmolarity of 2 osmol and exert a constant osmotic stress. Then the cells were harvested, washed, and the compatible solutes were extracted and quantified using NMR. And the results on this graph show that in stress conditions, Propionibacterium fraudenreichi accumulates more compatible solutes, differing with regard to the medium. As you can see, it is mostly trehalose, the blue bar, with accumulation with whey, whereas glycine betaine, GB, the yellow bar, is preferentially accumulated in EL. So the conclusion is that the lactose NPN ratio modulates the trehalose over glycine betaine ratio in terms of accumulation. Then we spread dried the cells harvested from the different growth medium and resuspended before spread drying in a 20% maltodextrin medium with a dextrose equivalent DE from 6 to 8. And as shown on the left figure, Propionibacterium fraudenreichi mostly accumulates GB when the medium contains NPN plus salt and trehalose in presence of lactose plus salt. On the right, the survival results showed that only lactose addition can enhance survival during spread drying via trehalose accumulation. So to our knowledge, this is the first report showing that in addition to triggering osmolar adaptation, it is possible in Propionibacteria to fine-tune the trehalose over GB ratio and thus to drastically increase survival upon spread drying. And in this work, we also worked on the freeze drying, and we showed that in the case of freeze drying, it was a glycine betaine that was very important for maximizing the survival. So in order to keep some time and give you a short glance of the last section, I will now illustrate the third topic and to share a bit with you about this redesigning of the powder production of whey permeate. So the conventional process scheme, as you know, for the production of whey and permeate powders consists in the concentration of liquid whey from 6 to 60% dry matter, lactose crystallization, and in batch often, and spread drying of the viscous concentrate. It is efficient to produce non-hygroscopic powders as far as you have a high level of crystallization. But the fact is that is a poor energy efficient process, and indeed the spread drying, as it is shown here in terms of specific energy consumption, SEC, is a very energy consuming step with regard to the value here that is like 12 times higher than the one of falling film evaporation. One solution, so it's overall 50%, 55% of the overall energy that is consumed during spread drying for only 4% of the water that is removed. So it is drastic. And the one solution to decrease the energy cost is thus to achieve a higher dry matter content at the outlet of falling film evaporators, but it results in the same times in higher viscosity, and it negatively impacts process and product quality. In particular, the higher viscosity makes it difficult to spray the concentrate. So our idea was to develop an innovative process based on thin film rotary evaporator, where the viscosity is controlled and lower because of a vigorous mechanical treatment that maintains a fluid flowing state at high dry matter. So what we propose is to replace the spread drying step by a super concentration from 60 to 80% dry matter in thin film horizontal and rotary evaporators, where a high shear is applied to the product, maintaining the concentrate in a fluid state, even at high concentration, such as 80% dry matter. The 80% dry matter paste, it is a paste, is then granulated into discrete particles by recirculation of the dry powders, and the granules are further dried in fluid bed dryers. So the feasibility of this patented process, which is named PST, was validated at the pilot scale, and we obtained satisfactory powder properties, non-significantly different from the controlled spread dryer powders, with energy saving that were estimated between 10 and 30% on the sole basis of the energy required for water removal. And besides, we expected additional savings as this kind of horizontal evaporators are much more compact than spread drying chamber, and the building savings were estimated at this stage at 30%. This pilot scale experiments showed the super concentration and the recirculation rate as critical parameters that determine the process performance in terms of energy savings and productivity. It was evidenced, in particular, that maximizing the extent of super concentration in rotary evaporator results in higher energy savings, logically, while lower super concentration was linked to increased recirculation rate and lower productivity. To get a better understanding of the limiting factor of this super concentration and granulation process, we conducted a joint PhD with Chegask in Ireland. Sorry, I have a cold. Up, sorry. This is not Brazil, yeah. Oh. And the main objective was to evidence the limiting factors, as I said, and we developed for this a lab scale model to mimic the novel process scheme using a rotavapor first for concentration and a food mixer for super concentration and granulation, a thermomix. The raw material was a skim milk microfiltration permeate, MFP, before unconcentrated for the superconcentration step by the rotavapor. So different analytical methods were used to characterize the process, such as rheology and shear cell measurement on the one hand, to characterize the rheological behavior and flow properties of the product during the superconcentration and granulation step. And on the other hand, the agitator power consumption of the food mixer was measured to provide further information about energy consumption and flow properties of the paste. And interestingly, experimental results on PMF, MFP, concentration evidenced a strong correlation between the apparent viscosity of the superconcentrated paste and the agitator power consumption. So this is very interesting because from the agitator power consumption, we can monitor the viscosity of the paste. Furthermore, the experiments showed that monitoring the agitator power consumptions was a reliable way to follow the evolution of the product behavior as a function of the dry matter. And this graph shows a critical zone between about 82 to 88% dry matter, which is characterized by a high energy consumption, as you can see, and where the super concentrated MFP paste evolved into a highly sticky mass with extreme resistance to flow. So on this graph, the point A corresponds to the onset of the cohesive phase, that is the dry matter beyond which the product transforms in this highly sticky paste. The point B corresponds to the cohesive phase, and the point C corresponds to the end of the cohesive phase, that is the dry matter where you obtain the discrete granules, particles that you are looking for, and for a minimum recirculation rate. And the picture on the right, on the bottom, sorry, show the aspect of this highly cohesive sticky mass. And these two pictures on the right show the aspect of the granulated powder at the end of the granulation for two recirculation rate. At the top for 1 to 0.8, one is the paste and 0.8 is the added powder. 0.08 ratio, sorry, the recirculation rate is too low, and you have always a sticky mass at the end of the process. And at the bottom is 1 to 1. The recirculation rate is okay, and you obtain the expected discrete granules powder. So the method was applied to different dairy streams afterwards, and whey permeate, as you can see, demineralized whey, and fat-filled milk and skim milk. And as you can see here, nevertheless, the product, you always see the similar trend with a point where you enter the highly cohesive phase, which is kept for a while, and then you go out of this highly cohesive phase for different dry matter contents, and that is what is interesting here. And what we can note is that a higher protein content, for example, here the skim milk or the fat-filled milk, shifts the highly cohesive phase towards lower dry matter content, means to the left, due to the higher water holding capacity of proteins, most probably, and limiting resulting viscosity. So to conclude, maximizing the super concentration is a key for obtaining energy efficient process. The onset of cohesive phase limits the super concentration, and the product composition influences the highly processed performance and the product functionality. So to conclude and give you some take-home message, so we showed first that colloid properties condition the skin formation and final particle properties. Then we highlighted that the tuning compatible solutes accumulation makes it possible to enhance the bacterial survival. And last, that the PST patented process control relies on the determination of the cohesive phase dry matter range and understanding, generally speaking. In the future, so we schedule extensive work on IMF formulation, and as process determines powder properties via particle intrinsic features, and can enhance probiotic survival during drying and in the dry state. We expect to develop new process, including this positive probiotics bacteria with success into IMF. So as such, the mother microbiota knowledge paves the road to gold standard. And finally, the research strategy to overcome the industry challenge, generally speaking, should address simultaneously hygienic, nutritional, and environmental issues. Identify the mechanisms at small scale, as I showed today. Then propose a scale-up strategy, and consider this multidisciplinary approach coupling physics, biology, and tomorrow, I hope, medical. So, merci, muito obrigado. And if you have any questions, I will be happy to answer. I hope I wasn't too long, but okay.